Good afternoon, and I'd like to welcome you to this second session of the day, where we hope to show you how the agency is involved in activities to address the problem of antimicrobial resistance, or AMR as we refer to it, and how the EMA is supporting innovation in the development of novel veterinary medicines, some of which could help to reduce our reliance on conventional antibiotics. So my name is Helen Dukes. I'm a former member of the CVMP, and I'm now working as a national expert in the AMR team at the European Medicines Agency. So just to give you a bit of background, according to a report from the European Centre for Disease Prevention and Control, AMR causes 35,000 human deaths every year in the EU, and also results in costs of around 1.5 billion euros annually to healthcare systems and in lost productivity. So clearly it's a very important issue. It's also one of the top 3 health threats faced by the EU as identified by the HERA, that's the Health Emergency Preparedness and Response Authority. However, much less is known about the burden of AMR on animal health and welfare. And AMR in animals will inevitably also affect human well-being through impacts on livestock productivity and food security. Likewise, although we know that AMR can be transferred from animals to humans via animal-derived food produce, through physical contact, and indirectly through the environment, further research is needed to clarify these transmission pathways and the extent to which antibiotic use and resistance in animals contributes to the overall burden of AMR on human health. Although several incentives have been proposed on the human side to encourage development of novel antibiotics with the potential to overcome existing resistance mechanisms, antibiotics with truly novel modes of action are likely to be reserved for human use in future. And on the animal side, strategies to manage AMR have focused mainly on stewardship and reducing antimicrobial use. With this in mind, the development of medicines with alternative modes of action to prevent, control, and treat infectious diseases in animals is becoming increasingly important. In the presentations today, we'll hear from Dr. Pote and Dr. Garcia del Blanco about the work of the EMA to encourage development of novel medicines. And we'll also hear from a researcher, Professor Guardabassi, on his practical experience of developing alternative products for treating diarrhea in pigs. But we'll start with Zoltan Khonshaghi, who will give us an overview of the activities of the EMA in relation to addressing AMR through the monitoring of antimicrobial consumption and encouraging responsible use. So just to give an introduction to Dr. Khonshaghi, he graduated as a veterinarian in Budapest and also holds master's degrees in toxicology and analytical chemistry. He worked as a laboratory analyst, a researcher, and a consultant in Hungary, Belgium, and Saudi Arabia before joining the EMA in 2014, where he now works in the veterinary division, leading the antimicrobial resistance team. So Zoltan, I'll hand over to you. Thank you.
Thank you very much, Helen. Good afternoon, everyone. And first of all, I would like to say that I'm quite honored to make this first presentation of this section. I have to say that antimicrobials and antimicrobial resistance is close to my heart. We have maybe a long-term relationship, which started probably at the university when, of course, I learned about it for the first time. Then I was practicing in small animal clinics, and I had the first time to meet in real life antimicrobial resistance when treating dogs and cats. Then in the laboratory, of course, I analyzed antimicrobials in food and in doing resistance tests, antimicrobial susceptibility tests, and then later joined the EMA. So Helen already mentioned the global importance of antimicrobial resistance, and today I would like to focus on the EU initiatives, especially on the European Medicines Agency role on the fight against antimicrobial resistance. During my presentation, I would like to focus on 3 main items, as you can see it on the screen. First, I would like to summarize the collection of antimicrobial consumption data, which guides policy and research. Then I will introduce how we promote responsible use of existing antibiotics. And then I would also like to give you examples how we provide advice to the European Commission. So let's start with our biggest success story, I must say. I will talk about now the collecting antimicrobial consumption data and will summarize how we do that. ESFAC stands for the European Surveillance of Veterinary Antimicrobial Consumption, and the European Medicines Agency holds this network, which basically collects harmonized sales data of antimicrobials for veterinary use in the EU and in the EEA. This project started in 2009. The first report was published in 2011. And since then, every year, we publish an annual report. On the right side, you can see the report which was published last year. And I can already tell you that this November, we will publish the 13th ESFAC report. In 2011, only 9 countries participated in this collection of antimicrobial sales data. But this year, when we will publish this report, 31 European countries' data will be included. I would like to highlight that this collection of antimicrobial sales data was always completely voluntarily. This is very important for risk assessment. I will show you how it is very useful for the trends analysis in antimicrobial consumption, and also important for monitoring of adherence to responsible use policies as well. On the bottom right of the screen, I included some blue boxes because as far as I know, these presentations will be available after this meeting. And if you are interested, you can just simply go to the slides and click on the link and read a bit more about this. So let me introduce a bit the trends in antimicrobial consumption. This graph shows a trend we are all proud of. When I say we, I mean all the member states who volunteered to provide this data, and here at the EMA. It shows a trend between 2011 and 2021. Sales data from 25 European countries are included. On the top of the figure, you can see a line which clearly is decreasing. And I can summarize this that overall between 2011 and 2021, 47% overall decrease was observed. This is what we call primary indicator, the most important indicator, how the sales of veterinary antimicrobial products decreased during this period. On this figure, you can also see some bar charts as well, and you can clearly see that these numbers are also decreasing. These bars represent 3 very important antibiotic classes, which actually classified by WHO the highest priority critically important antimicrobials, namely the 3rd and 4th generation cephalosporins, polymyxins. Maybe you know colistin better, the colistin antibiotic, which is part of this polymyxin antibiotic class, and fluoroquinolones and other quinolones decreased as well. I won't repeat the number of the decrease, what you can clearly see on the screen, but I just would like to highlight one thing. 80% decrease was observed for colistin, for polymyxins, And I think this really clearly shows that policy initiatives work. Let me introduce you some important documents which were published by the European Commission. So in 2017, the Commission adopted the EU One Health Action Plan against AMR. And a couple of years later, in 2020, the Farm to Fork strategy was published, which renewed emphasis on antimicrobial resistance. This Farm to Fork strategy had a couple of targets until 2030 for sustainable food and production. Today, I just would like to highlight the third one, which is seen on this slide as well. So the goal is, which was set by the Commission, that until 2030, the reduction of the sales of antimicrobials for farmed animals and in aquaculture should be 50%. This is our goal. Where are we now? So to reach this goal, we still have a couple of years. We have to reach it by 2030, and the reference year is 2018. 118.3 milligram per PCU was the baseline, and we need to reach 59.2 milligram per PCU. For those who don't know, PCU is a technical unit of measurement where basically one PCU unit is equivalent of 1 kilogram of animal biomass. So between 2018 and 2021, we already reached 96.6, which means that out of 50, 18% decrease was already achieved. So I think that's a really good achievement I wanted to highlight today during my presentation. So what I explained so far is all thanks to our surveillance network, to SFAC. Again, thanks to all the member states who participate and voluntarily provide this data. Again, I would like to highlight voluntarily because this will change. This will change next year. And this morning, you already heard some presentations about this. I also need to refer to the Veterinary Medicines Regulation 2019/6, because according to this regulation, the collection and reporting of data on the volumes of sales per veterinary antimicrobial products will be mandatory. The collection and reporting of data on the use per animal species will be mandatory as well for both veterinary and human. So again, as it was voluntary before to collect sales data, from next year, it will be mandatory to collect the sales and the use of antimicrobial products per animal species. This will be a big change. Also, my colleagues are quite busy with the development of an IT system for data reporting with the help of member states representatives as well. And this IT system is already available for testing since January 2023. It's important also to mention that this will be linked to the Union Product Database for veterinary medicines and also to the Human Article 57 database. To help all this work, what needs to be done in the whole Europe, a new working group was established. The name of this working group is the European Sales and Use of Antimicrobials for Veterinary Medicine Working Group. In short, we just call it AVANT. So I think the SFAC name is already quite well known for those who work in this area. I think in the future, you will remember and you will notice AVANT more easily or more frequently. So what are we doing with all this data? Yes, we collect it. Yes, we analyze it. Yes, we publish it. But there are so many things to do what we can do with this data. As they say, data is the most important, the biggest value what we can have. Let me give you an example where we use this data and we'll use in the future as well. So the Joint Interagency Antimicrobial Consumption and Resistance Analysis, or the JIACRA report, is a good example where the European agencies work together, share the data, what they collected with their surveillance network. This JIACRA report is, again, a great collaboration between 3 EU agencies. The European Centre for Disease Prevention and Control, ECDC, which is in Stockholm, the European Food Safety Authority, which is in Parma, and of course the European Medicines Agency. What is this JIACRA report about? This JIACRA report analyzes the associations between antimicrobial consumption, antimicrobial resistance in humans and food-producing animals. Already 3 JIACRA reports were published. The last one was published in 2021. And I'm happy to tell you that currently we are finalizing the 4th JIACRA report, and it will be published by the end of this year. Again, if you are interested in more information on these reports, please click on the bottom right of this slide when this will be shared with you. The first part of my presentation was really about the collection of data, and I would like to give you an example how we provide guidance on the responsible use of existing antibiotics and how we promote it. The CVMP's Antimicrobials Working Party published some guidance documents, reflection papers like aminopenicillins, aminoglycosides. Again, you can find these reflection papers on our website. Another good example for good collaboration between EU agencies is the RONAFA report, which is a collaboration between EFSA and EMA. And it's a scientific opinion. And if you are interested how to reduce the use of antimicrobials in food-producing animals, if you are interested what these 2 agencies recommend, I also would like to kindly encourage you to read this report. But in this slide, I would like to promote another advice, which is about the categorization of antibiotics in the EU. I'm pretty sure that you all heard about the categorization of antimicrobials, antibiotics by different international organizations, like the World Health Organization, the WHO, categorizes the antimicrobials according to the importance of human health. The World Organization of Animal Health, VOA, previously formerly known OIE, categorizes the antibiotics by the importance of animal health. This AMEC, which stands for the Antimicrobial Advice Ad Hoc Expert Group, this AMEC categorization, which is the European Medicines Agency's categorization, aims to balance human and animal health needs and public health with public health considerations. We have 4 categories, as you can see on the screen. Category A contains those antibiotics which are not authorized in veterinary medicine, but authorized in human medicine. Category B is a lower category with those antibiotic classes which I presented earlier, namely cephalosporin, quinolones, 3rd and 4th generation cephalosporins, quinolones, and polymyxins. Category C and category D, and so on. And this categorization provides guidance to the veterinarians because it supports their decision-making, which antibiotics to choose from. So what we suggest that, of course, if it's possible, choose something from category D class. If it's not possible, then category C, B should be the direction. You can ask why, what else we can do with this categorization of antibiotics. It also, for example, assists the development of local treatment guidelines as well. Again, this is something which I would like to kindly ask you to click on the slide and download our nice infographic, which is translated to 23 EU languages. And I would like to suggest to all the veterinarian colleagues to print this out, put it on the wall on your clinic, because I think it gives you a nice overview of all the antibiotic classes which are used. Also, those antibiotics which are available only as human products, but could be used off-label as well. And finally, the third point what I would like to introduce to you is the support of the European Commission. This was also a topic which was mentioned this morning. EMA provides scientific advice to the European Commission for the support of the veterinary medicines regulation. Let me give you two examples. The first one is the antimicrobials, which are reserved for human use only. What does it mean? I think it's now quite obvious, you can tell. So there is a list of antimicrobials which will never be used in animals. Those antimicrobials can only be used in humans. And it also applies to imports of animals and their produce as well. Almost 400-page long scientific advice was prepared by the European Medicines Agency with the help of the working group. This working group had experts who were veterinarians, microbiologists, medical doctors specializing in infectious diseases, external experts. We also had an open call to gather information, and the group analyzed all the antibiotic classes individually, and also this evaluation was based on the best scientific evidence currently available. This advice recommends 11 antibiotic classes, 6 antibiotic substances, and 18 antiviral substances to be reserved for humans. I won't give you now all the list, but just to give you an example, carbapenems is a last-line group of antibiotics which is mainly used in hospitals for the treatment of patients with multi-drug resistance gram-negative bacteria. So these carbapenems are reserved for human use only. As you can see, there are antivirals as well, for instance, favipiravir or rimantadine, which are important antivirals against influenza. This is the strictest measure in veterinary context to address public health AMR issues. This advice was adopted by our committee, by CVMP. It was sent to the European Commission, consulted with the member states. It was debated at the European Parliament, and now we can proudly say that it became an implementing act, it became legislation. The second example, which I would like to give you, how we support the Commission, what type of scientific advices we provide to the Commission, is a list which is complementary to the previous reserves for humans. We can all agree that not all antibiotic classes are available for veterinarians to treat animals. It's just so clear. We don't have all possible antibiotic products available for all species and for all indications. So that's why many times or sometimes veterinarians need to use antibiotics outside the direction on the product label. And that's why we received this mandate from the Commission to develop a list where outside the direction on the product label, which will be not possible for certain antibiotics, or it will be restricted use. Again, a working group was established to provide this advice, and the latest scientific advice information literature was evaluated to provide this large document. Existing recommendations, official reports, and surveillance data from all EU agencies we work with were considered. More than 700 supporting references were evaluated. So in this advice, we provide certain recommendations, conditions for use. Just to give you an example, we provide certain classes to have antimicrobial susceptibility testing, or those could be used only in individual animals or for certain diseases, or restriction in the route of administration, for example, topical only. This advice was also sent to the Commission recently, and there will be an implementing act as well in the future. So now I reach the end of my presentation, and I hope you can understand my statement that EMA plays a vital role in the global response to AMR through the One Health approach. During my presentation, I try to summarize how we collect antimicrobial consumption data to guide policy and research. I introduced SFAC and the new AVANT. I give you an example how we promote responsible use of existing antibiotics. Here I give you an example of the AMEC categorization, how we provide advice to the European Commission by two scientific advice, which I just mentioned. But there are a lot more what EMA is doing. For instance, we support the development of new medicines and treatment approaches, and today you will hear more about this. And finally, I also would like to mention another thing, that interagency, international cooperation, also work with academia, with member states are really essential to share our experience and knowledge and work together in the response to threat, the antimicrobial resistance. Thank you very much for your attention.
Thank you, Zoltan. And I think the format for today's session is that we'll take questions all together at the end of the 4 presentations. So without hesitation, I'll move on to Professor Guardabassi. So Luca Guardabassi graduated as a vet from the University of Pisa and completed his PhD in veterinary microbiology at the University of Copenhagen, where he has been professor of One Health AMR since 2012. His research has focused on the epidemiology of multi-drug resistant bacteria of clinical relevance in human and veterinary medicine. Luca is the vice chair of the Scientific Advisory Board in the Joint Programming Initiative on Antimicrobial Resistance, that's the JPI-AMR, and he's a pioneer of antimicrobial stewardship in veterinary medicine, being involved in several national and international committees for the development of practice guidelines for rational antimicrobial use in animals. So, Luca, I'll hand over to you. Thank you.
Thank you. And thank you to EMA for giving me the opportunity to present this project in front of this highly qualified audience. This is an example of about development of alternatives to antimicrobials supported by the European Union. And you can see some details about the project on this slide. I would like to remark it is a typical example of multi-actor project where not only researchers but also professionals from different sectors are collaborating together to, in this case, develop alternatives to antimicrobials. And it is, as part of the consortium, there are 5 universities, 4 industries, 4 SMEs, and 1 large veterinary organization, which is the Federation of Veterinarians of Europe. So for a total of 14 partners from 9 countries. And I became familiar actually with this terminology, technology readiness level, with this project. It is something that is used about development of commercial products and bringing products closer to the market, in particular innovation actions, which is the instrument, the financial instrument that is supporting this project are supposed to produce new or improved products and bring them, in this specific case, from TRL level 5 to TRL level 7, where 5 is basically testing the alternative in an animal facility, and 7 is testing a prototype of the alternative in an operational environment, which means a farm where pigs are sent to the slaughterhouse. And we decided, our strategies was to decide on a single animal species and a single disease. And the reason was that pigs account for the vast majority of antimicrobial consumption in the livestock sector. And here there are some data you can see in the graphic reporting the biomass production of different animal groups on the left, and on the right the amount of active compound of antibiotics in Denmark. You can see that although pigs represent less than half of the biomass, they account for approximately 80% of antimicrobial consumption. And most of these antibiotics are used during the weaning period. The weaning, for those who are not familiar with this term, is the time where the pig moved from milk-based diet from the sow to solid feed. And this is an extremely stressful time for piglets in intensive production systems. You have to think that the natural, in nature, sows would wean normally their piglets around 14, 17 weeks. And in intensive production, this is reduced to 3, most countries 3 weeks, in some countries like the Netherlands, as much as I know, 4 weeks. So these animals are extremely fragile, and they have to face a stressful condition because of the change in the diet, but also a social stress because they are mixed together with piglets from other litters. And this is extremely stressful, and the stress results in a susceptibility to specific pathogens that cause diarrhea in these animals. And the most important of this pathogen is an Escherichia coli strain that is producing toxins, but this is less important in this context. What I would like to remark by this slide is that there is actually, this is a very good example where there is need for new antimicrobial solutions, because there are some vaccines that are available, but for a number of reasons, it is extremely complicated to develop an effective vaccination program due to the age of these animals, but also the complexity of the disease, the variety of enterotoxigenic E. coli that can be involved. But also there are studies showing that these enterotoxigenic E. coli they're not the only pathogenic pathogen involved in the etiology of the disease. So there were 2 classes of antimicrobials that were extremely effective in controlling this disease. Zinc oxide, which has been banned to prevent the transmission of MRSA, methicillin-resistant Staphylococcus aureus, from livestock to humans. And colistin, that has been mentioned in the previous presentation, that has been restricted, especially in Denmark, but all over Europe, because of the risk of transmitting colistin resistance in gram-negative pathogens. And then there is, shown in the graphic on the right side, there has been an increase in resistance to the antibiotic that is used as a first choice for treatment of this disease. which is neomycin, an antibiotic that is actually very little used in human medicine, primarily as a topical product, and is used orally in pigs because it has a very low oral bioavailability, so it concentrates in the intestinal tract and is poorly absorbed, and therefore reach very high concentration at the site where enterotoxigenic E. coli proliferates. And as it is shown in the graphic, when neomycin was reintroduced in Denmark in 2017, the prevalence of resistance in clinical isolates of this pathogen was relatively low, around 10%. And after we have started using this drug, we have seen an exponential increase in resistance. And these are data from pathogenic strains isolated from diseased animals. And the last data from 2022 indicated that ETEC have reached about 40% resistance to neomycin. This data may be biased because not all veterinarians are sending samples to the diagnostic lab. But this roughly tells us that at least 1 out of 3 infections, outbreaks caused by ETEC is resistant to the first choice antimicrobial. So there is need for solutions here. And this is why we decided to focus on this disease. And even though we focus only on one single animal species and one single disease, we decided to have a very broad and comprehensive approach in terms of types of solutions. So the AVANT portfolio includes 4 categories of alternatives, the so-called gut microbiome modulators, which include symbiotics, a mix of probiotics and prebiotics, and a specific on-farm protocol for fecal microbiota transplantation. I will touch upon this in the next slides. Then some products that fall more under the category of veterinary medicinal products, in particular some polymers and mixes of phages that target this pathogen, enterotoxigenic E. coli. Immunostimulants, so drugs that act on the immune system of the animal, and we have tested both oral and systemic products. And finally, a 4th work package was dedicated to feeding strategies targeting either the sow or the piglets. So the aim of the project was to develop and test these alternatives and see what is their impact actually on antimicrobial use in pig production. And this is a little bit the overall structure of the project, showing the 4 scientific work packages where these products, these alternatives were developed and tested in preclinical studies. And now we are actually at the stage where we move from the animal facilities to the clinical trials that will be performed in 3 countries, including the Netherlands, Denmark, and France. And of course, based on the results of the preclinical studies, we made the selection. We could not test all the interventions, and for some of them, there were also legal constraints in using them in real commercial farms. So we did an evaluation of the results, and 2 interventions were selected based on their efficacy. One is fecal microbiota transplantation, which I will cover in detail during the next slides. Developed by the group of Professor Thomas Thumann at the University of Copenhagen. And the second one was a fiber-rich diet of the piglets, developed by a small medium enterprise in the Netherlands, Scottish Feed Research. And as I said before, now we are in the most exciting phase of the project, where we test these interventions and not only assess their efficacy in preventing and controlling the disease, but also the cost and therefore the sustainability of the interventions and the effect on antibiotic use. So I will start from some preclinical data regarding fecal microbiota transplantation. And before presenting the data, the overview of the data, I should mention that this is a protocol that is performed on farm, meaning that the donor of the feces are recruited within the farm where the protocol is applied. So there is, in such a way, we avoid the risk of spreading pathogens from one farm to another. And initially, we tested 3 different types of fecal transplant: the total transplant, the gastric intestinal transplant, the gastric transplant, and also for the fecal transplant, we tried a filtrate to filter, because there is some evidence from human studies that filtered feces may actually have an equal or even better effect compared to intact feces. And I should also mention that fecal microbiota transplantation, for those who are not familiar with this procedure, is currently used in human hospitals for control of specific intestinal disorders, especially those associated with Clostridium difficile. So when we tested these 3 inocula in piglets, single-housed piglets, at the animal facility, we were not particularly excited about the results. You can see in this graph on the y-axis the percentage of animals displaying diarrhea, and on the x-axis the days of age. And as I said, the weaning starts at around 3 weeks, so day 21. And we could see a significant effect only for the fecal filtrate around day 28 and 29. This was statistically significant, but as you can see from the graph, we could still see around 20% of piglets displaying diarrhea at day 28. So it was a reduction in the number of diseased animals rather than a complete prevention of the disease. And this second graph is reflecting the same, is just expressing, instead of the number of piglets displaying diarrhea, the consistency of the feces in the 3 treatment groups in comparison with the control group. And again, we saw a significant difference at day 28, meaning that the feces were more solid in the piglets treated with the filtrate. So we decided to move on and test this protocol in an environment that is closer to a real farm, also because these piglets were artificially inoculated with the pathogen. I forgot to say that on day 24, as you can see on the first graph in the middle. So we moved to an experimental farm where post-weaning diarrhea occurred naturally. And the filtrate was administered during the first 6 days, so when the piglets were still together with the sow. And as you can see here, the weaning was implemented at 4 weeks, day 28. And periodically, there was a clinical and fecal score of individual animals that was performed throughout the study until the animals were euthanized at day 40. And in this case, the results were much more promising compared with those obtained under controlled conditions. So we saw that the daily occurrence of piglets displaying symptoms of diarrhea was significantly lower in the treatment group compared with the control group. And here, the number of asterisks are only indicating the level of statistical significance. But as you can see, throughout the post-weaning period, the fecal filtrate was able to reduce significantly the number or the percentage of piglets that were affected by the disease. And of secondary importance in the first days of life. So when the piglets were still treated with the filtrate at day 6, we could also see a lower percentage of piglets displaying neonatal diarrhea, which is the second most critical phase for antimicrobial use in pigs after weaning. And this graph is showing the effects of the treatment on mortality. Again, there was a significant reduction in the number of pigs that died or were euthanized because of the severe clinical conditions that they presented. This, because of the study design, antibiotics were not used during the study. So when the animals were had displayed the severe illness, they were euthanized. So there is very good news in this graph that by using this approach, which is relatively inexpensive, we can actually reduce from around 30 to 20% the mortality in piglets. This is good news, but at the same time, we have to realize that without using antibiotics, we would have to lose about 20% of the production at this stage. So then we had to move to the real farms. As I mentioned before, fecal microbiota transplantation was selected for the clinical trials. But 2 of the countries, Denmark and France, denied the authorization to perform the clinical trials because of public health safety. I have to mention that there are presently some regulatory constraints for the marketing of this product, and even for testing the product in pigs that are supposed to go to the slaughterhouse. And this is because it is not a feed additive, and according to the current EU regulation, feces or any derivatives of feces cannot be used to feed animals. And is not obviously a medicine because its content is not standardized, and even the mode of action of this fecal filtrate is not elucidated. So I hope that this project will also help to start a process, a dialogue with the stakeholders. And I forgot to mention in the beginning that AVANT is a multi-actor project, not only because of the composition of the consortium, but also because we have the pleasure to have in our external advisory board different agencies, including EMA and EFSA, and different associations representing the stakeholders involved in this fight against AMR in livestock production, like COPA-COGECA representing the farmers, FIFAC representing the feed industry, and the animal health industry as well. So I hope that by this project, at least we will be able to set a path for future projects, so that it is known what can be done and what are the requirements for testing these products in farms. There are also some technical challenges that should not be overlooked. We haven't actually paid a lot of attention to the quality of the donors. We have just selected some healthy animals, but much more research can be done to select the optimal donor feces. We have seen that filtrates are more effective based on our data than the actual feces. But this requires some on-farm equipment like centrifuges and filters that are needed for the implementation of this protocol on farm. And then finally, also handling the piglets individually is not particularly easy, and is also a little bit stressful for these animals, of course. Then I will mention a survey that we have conducted among the end users, or at least the 3 categories of end users of these products, pig farmers, pig veterinarians, and consumers, to understand the level of acceptance of different types of alternatives to antibiotics. And this was conducted in 2021. We subcontracted a specialized company, PNM, that is based in Germany, which conducted a telephone survey on a large sample of respondents belonging to these 3 categories. And this was conducted in 5 countries, which include some of the most important pig producing countries in Europe, and also culturally, we try to cover as much as possible the diversity within our continent by including countries from Northern, Southern, and Eastern Europe. And this is a long publication is on the way, and so you will soon get access to the full results. But if I should highlight the most interesting ones, is that all 3 categories, including farmers and veterinarians, have a better perception of the traditional measures like vaccination, biosecurity measures, compared to more innovative products like fecal microbiota transplantation and phage therapy, meaning that in addition to the regulatory challenges that we are facing, we are also facing some cultural challenges. So part of the work will also be to raise awareness about the efficacy of these products among the professionals, and maybe also to emphasize the safety of these products from both public and an animal health perspective. And then a second element that I have decided to highlight here is that about 70% of the consumers on average across the 5 countries said that they are willing to pay 10% or more to buy food that is not produced with animals treated with antibiotics. And here we have to be careful in judging this result or interpreting this result, because there are actually real data showing that consumers are not particularly attracted by food products that are more expensive, even though they are not produced with antibiotics. And a good example of this is that in Denmark, the Danish pig industry decided to start a production, an antibiotic-free production, some years ago. And this is actually going to be shut down next year. At least this is what I have heard, because it doesn't seem to be a profitable market. So some take-home messages and maybe some point for a discussion at the end of this session. First of all, let's be aware that based on the state of the art, we do not have alternatives that are able to completely replace antimicrobials. It seems that some of them may be useful in reducing the use of antibiotics, but we still need antibiotics for curing the diseased animals. And in this respect, it would be important. So far, at the European level, we have using this benchmarking approach, and we have just been told that the latest reduce the overall antimicrobial consumption by 50%. But if this is the situation, we need to understand when the reduction will actually start affecting animal welfare. The graphic on mortality was quite clear. If we didn't use antibiotics in that farm, 20% of the piglets would have died regardless of the alternative treatment. This is one point. The next one is that we have been funded to test by the European Union to test alternatives to antimicrobials, and according to the regulation of the European Union, we are not allowed to test them in clinical farms. So we need to align a little bit the regulation to the needs of this society. There is a big societal pressure to combat AMR and reduce antimicrobial use. So as I said before, I hope that, first of all, we will have this tested, the fecal microbiota transplantation in this country, in the Netherlands. So I hope that the results of this clinical trial will convince more countries to accept testing, more trials that will be, of course, needed before this approach can be marketed or at least legalized at the European level. And the last point is that, as you can see, there are also some cultural barriers that we have to face, not only among consumers, but also among veterinary professionals and farmers. And altogether, my point here is that we need a holistic approach that is involving political decisions and also public acceptance. And if we really want to avoid the routine use, metaphylactic use of antibiotics in specific production systems like the intensive peak production, this require also a change of the production system. And the change of the production system will imply an increase of the cost of the products in the supermarkets, in the market. Personally, I would be in favor of paying even 40–50% more meat. I find it quite unfair to the animals that we pay 1 kilogram of chicken as much as a bottle of Coke. But this is, of course, a personal opinion that goes beyond my expertise as a microbiologist and infectious disease specialist. I would like to close the presentation by acknowledging, of course, the European Union for supporting this important research program. And also, I forgot to say that part of the data that were presented were co-financed by the Independent Research Fund Denmark and by the umbrella organization of livestock production in Denmark, SEGES. And last but not least, you can see the faces of Christina, who performed, was the scientist responsible for the performance of the experiment, and Thomas, who was the supervisor. Thank you very much for your attention.
Thank you, Professor Guardabassi, and it's very gratifying to see that despite all the challenges, there's still a lot of research going on into quite a wide variety of alternatives. So the next presentation will be from Jacqueline Pote, and this is a presentation on the work of the CVMP's Novel Therapies Working Party. So Dr. Pote graduated from the University of Utrecht and has worked in farm animal and companion animal practice. Her PhD work was focused on canine leishmaniasis disease models and vaccination. Jacqueline was a project leader in the biological R&D department of a large pharmaceutical company for 12 years before she joined the veterinary department in the Dutch regulatory authority. She's now a CVMP member, and she's also a member of the CVMP's Scientific Advice Working Party, and she chairs the Novel Therapies and Technologies Working Party. So I'll hand over to you, Jacqueline. Thanks.
Thank you, Helen, and thank you for the organization for inviting me to speak to you on one of my favorite subjects, which is the veterinary novel therapies and technologies. For the remainder, I will refer to this as novel therapies. Otherwise, I won't stay within the designated time. So I will not be complete on this subject because that's just not possible. Because, well, as it says already, to boldly go where no one has gone before. But I also will not be able to tell you everything on what is going on, but I will give you some examples of novel therapies. And I will try to indicate where these novel therapies may be of help in reducing antibiotic use. So novel therapies are specifically mentioned in the veterinary regulation, which is the law that has been mentioned before today several times. So what it says is that this is any technology not currently used in veterinary medicines. And this regulation also gives specific examples, which are nanotechnology, bacteriophages, gene therapy, cell-based therapy, and others. So those are the ones also not currently used and that we not currently know of yet. So I will take you through some of these at warp speed, I'm afraid, but we will see. So for the nanotechnologies, this is a very wide area of applications. You could think of, well, any substance at nanometer scale. That means that and one or more dimensions of a substance will be at nanometer scale. So, and for that reason, they will have particular properties that are other than for the same substance that is not that does not have nanometer dimensions. So it changes their properties. So you could think of nano antibiotics, where nanostructures can act synergistically with antibiotics. So there's one there that may help reduce the use of antibiotics as such. Nanoparticles as delivery systems or adjuvants for vaccines. Again, since vaccines are have the potential to reduce antibiotic use, again here, nanotechnology may help reduce antibiotic use if they can help us produce more effective vaccines. And then nanoparticles can also be used for targeted drug delivery systems and also a bit more remote and not necessarily a veterinary medicinal product, but nanosensors may be a help because they can help earlier or more accurate detection of disease or infections. So we go on to the bacteriophages. We've heard them before today. These are particular viruses that can infect only bacteria, and they have the ability to kill those bacterial hosts. They are generally highly specific, so they infect only one bacterial species. And as you see on this picture, the little space invaders here, the red ones, that are, these are depicting the actual bacteriophages. They look very fancy, I think. So with respect to bacteriophages, what is interesting about them is that bacteria and phages are in an evolutionary arms race. This is a picture of the Red Queen from Alice in Wonderland, who has to keep running to stay in the same place. And that is also what bacteriophages and bacteria are doing. So an infection with bacteriophages forces bacteria into defensive actions. And I have to note here that these defensive actions may weaken them in other areas, which could make them, for instance, more susceptible to antibiotics, which is a good thing. But these bacteria adapt to combat the infection by the phages. And then again, the phages adapt to be able to attack the bacteria again. And so they keep on going. And for that reason, while bacteriophages are already applied in humans and in animals with some success, this is often only to specific patients and for a limited time. But we think that wider applications may be possible. The problem with the bacteriophages and the bacteria is that they adapt so quickly, as I explained before, which makes it a little bit different. And also that sets them aside a little bit from what we are used to for veterinary medicines. So a complete cure of an infection may not always be the goal. It may not always be possible, probably not. But for instance, you could also think of decontamination using bacteriophages of the environment or of animal feed or food. for humans. And it may also be possible to use them in conjunction with antibiotics. As I said before, sometimes phages attack the bacteria in an area, which makes them then more susceptible to antibiotics. So they could be very useful there. Then gene therapy for veterinary medicine. There are some more and some less obvious potential future targets. One of them, those that are, I think, more obvious, I've tried to list here. So there could be applications in inherited diseases or genetic defects generally, for instance, for ocular diseases or metabolic diseases. There could be applications in wound healing, so that would enhance or speed up wound healing. Well, there may be a small opportunity to reduce antibiotic use if you do use antibiotics in wound healing. Then for fertility regulation, that had not much to do with antibiotic use, I think. And for chronic diseases in animals like osteoarthritis, diabetes, allergy, and renal and ocular diseases. And again, there for allergy, if you talk about skin allergy, there is generally quite a bit of antibiotic use there. So again, if we can cure that in other ways without the use of antibiotics, that would be good. So, yes, I am the chair of the Novel Therapies and Technologies Working Party, as you've heard. Why are we there? Well, as I said, there is a mentioning of novel therapies in the veterinary regulation. I've quoted a little bit of it before, saying what it is. And this is another quote from the regulation where it says, taking into account the specificities of novel therapy products, specific requirements additional to the standard requirements for evaluation of quality, safety, and efficacy may be appropriate. So some aspects of novel therapies may be very different from what we know. Nanotherapies is a good example where the scale of the substance changes its properties. So we may need additional testing to assure quality, safety, and efficacy. And the Working Party tries to provide guidance for that aspect. So where do we need different or additional data for this type of therapies? So how do we do this? Well, so the CVMP instated this Novel Therapies Working Party. So we try to develop guidelines. So the Working Party decides what guidelines are going to be developed, obviously in conjunction with CVMP. We do also horizon scanning to see, what's happening in the field, what sort of therapies and technologies are being developed that are relevant for the veterinary area. And we supervise the work of the operational expert groups. So operational expert groups consists, well, they have the function to write these guidelines on these specific topics. So for each topic that we address, there will be a different operational expert group, and it will contain experts. They can be from the veterinary and the human field, from academia, from institutions, from national authorities. The operational expert groups that we have so far also had members from, for instance, the FDA and the EDQM, and EFSA, so all institutions that we've heard of before today, and they can be there as observers, and they also bring their expertise. So the trick in the operational expert groups is to bring together these two worlds, because, well, the academic world where these developments are going on speaks a totally different language from the regulatory world, and well, within the operational expert groups, I've seen it happen. We actually managed to communicate quite well and in the end come up with guidance. And this guidance is then approved by CVMP and then published. And it can be used by both developers of novel therapies, but also by the authorities that are there to assess the dossier for such a medicinal product to help this process to be consistent. So what we've done so far, the Novel Therapies and Technology Working Party is a very young working party, but so far we've managed to produce and publish one guideline on cell-based therapy products. So that would be stem cells for use in veterinary medicine. And then the one on the quality, safety and efficacy of bacteriophages as veterinary medicines is in the final stages prior to publication. And there is one under development, which is on the safety of nanomedicines. So, well, thank you very much. This was my contribution.
So thank you, Jacqueline. And it was very interesting to hear the practical applications for some of these what sound like science fiction type products. So thank you for that. The next presentation that we have is from Dr. Garcia del Blanco, and she is going to give us an update on the opportunities in the new legislation and how the EMA is taking these forwards in relation to developing novel medicines. Noemi Garcia del Blanco is a veterinarian with extensive postgraduate qualifications in the field of microbiology and infectious diseases. She's contributed to the development and regulation of vaccines and other biological veterinary medicines, both in industry and in regulatory bodies. And she is now the head of the Biologicals and Emerging Therapies Service in the veterinary division at the EMA. So when you're ready, Noemi, thank you.
Thank you very much, Helen. Good afternoon. I'm very pleased to close this session. And I hope, and the intention is that my presentation is going to include everything that has been presented this afternoon, and bring it together to look at how we can, what we are doing now, and we can do in the future to promote the authorization of those products that can be alternatives to antimicrobials. Yeah. So this is what is going to include. What are we referring when we stay promoting the authorization of alternatives to VMPs, to veterinary medicinal products? And that is going to include the opportunities created by the current regulation, not new anymore, international cooperation and the existing support to developers and applicants, and then look at the challenges that are presented to the authorization of alternatives, and we have heard some before, and how we can overcome those challenges moving forwards. And first is promoting the authorization of alternatives to antimicrobials. Why is it important? What we are referring is to support and foster, we would say, the development of an authorization of those VMPs which can provide an alternative treatment approach to either the use of antimicrobials in animals or the need for the use of those by preventing or controlling infectious diseases. So I have a couple of examples there on the first case, as Jacqueline was already telling us about, bacteriophages. And for the second, a very clear example of a product that could reduce the need for antimicrobial use using as a preventative approach would be vaccines. And how do we do this? Well, we have to recognize the importance of this group of products as alternatives, and therefore adopt a proactive approach to promote their authorization. And I'm going to highlight three different areas. The first is going to be what is in the current regulation that is going to ensure that we have the available regulatory framework and the guidance in place for authorization of these products. I'm going to refer briefly, because you heard everything this morning about that, international cooperation as exchange of information and also the advice and support that we have in place for developers and applicants seeking to authorize these alternatives in the EU. So as I said, the first one would be I have new regulatory landscape is relatively new. So the regulation that we have currently in place and what it includes to stimulate innovation and increase the availability of these, veterinary medicinal products overall, but in particular this. And I want to highlight a couple of points. One is the requirements for a specific type of marketing authorizations. And I have included a list in the slides, so that would be applications for authorization in exceptional circumstances, for limited markets, for vaccine antigen master file, for vaccine platform technology, for multi-strain dossier, and for novel therapies. I'm going to look at a couple of them in a couple of slides. I also wanted to mention that this regulation also includes and differences between different types of products, which would be biologicals other than immunologicals, and examples, stem cells, phages, the immunologicals, for example, vaccines, and products other than biologicals, that would be traditional pharmaceuticals. And I just refer to this because that means that each of these categories have specific requirements in the regulation or in the annexes to the regulation, so they provide the regulatory framework for authorization. Yes, as I said, I'm going to refer to a couple of them because I think they are quite interesting for this forum. The one is applications for authorization for limited markets, which are a specific type of authorization according to a specific article in the regulation that may be granted for limited markets in the absence of comprehensive safety and or efficacy data, when the applicant can demonstrate that the product is indeed aimed for a limited market and the availability, the benefit of the availability outweighs the risk of this lack of certain data. And what is a limited market? Well, is either a product for the treatment or prevention of diseases that occur infrequently or in limited markets, in limited geographical areas, or products for animal species other than cattle, sheep, meat production, sheep for meat production, pigs, chicken, dogs, and cats. For instance, the fluffy chinchilla in the photo, that would be a minor species, that we could say. And I said, I think these are quite, interesting for the point of view of the use of antibiotics, because sometimes the availability of products for those specific markets is not the best as desired. The next one I wanted to highlight, obviously, is applications for novel therapies. Jacqueline has touched on this, so I'm not going to go there. Just to say that, again, we have the different type of products because novel therapies could fall under each of these categories. And then there's some flexibility for specific types of products, depending on the type of product. Either additional requirements could be relevant, but there could be also some flexibility on the data at the time of authorization, and therefore it could be that implementation of post-authorization measures are considered on case-by-case basis, depending, as I said, on the type of product. I think we don't need to touch on novel therapies anymore because Jacqueline has covered everything. The second point I wanted to highlight is the international cooperation and exchange of information, because in this area is very highly important. You heard quite a lot this morning about international cooperation overall. Just to say that more specifically in the development of alternatives to antimicrobials, the agency exchanges information with specifically FDA and the USDA. And of course, collaboration with other European agencies like the European Food Safety Authority. And there's some specific activities for the discussion or forum for the discussion of the requirements for authorization of novel therapies, for instance, in the TADFAR, that I think it was also mentioned this morning. So it's an international forum where the challenges and the requirements of this type of products are discussed. So what is available at the moment to support the development or the developers and applicants of this type of products? And this is hopefully, and I have a graph in the next slide where you can see the different measures in place according to the storyline of the development of a product, starting with early inquiries, a schema, which is general place to send inquiries to questions to the agency, which is easy to find in the web page of the agency. The next one would be Innovation Task Force, ITF, and this is also a forum for discussion where experts from the European network meet and colleagues from the agency, and is at an early stage of development where aspects of legal, regulatory, and scientific aspects can be discussed. It's a bit more, it's very early in the development, as I said, it's a bit more general, or it could be product specific. The next step, I would say, would be scientific advice. So it's a bit later in the development of a product. Of course, usually pre-submission, it could be during the evaluation, but usually more valuable when it's before submission. And the next step would be pre-submission meetings where the product is in the final stage of the development. Most of the work is that, but there are specific questions. The applicants are very welcome to discuss them, and we, the agency, organize that kind of meetings. Then, of course, we have in place guidance and guidelines on the agency website on procedural, regulatory, and scientific issues that are produced by aided agencies, more in procedural aspects, or through the CVMP, or the CVMP through the different working parties, more scientific advice. And I just highlighted there in the slides the Immunological Working Party, working in obviously producing guidelines for vaccines and immunologicals, and then NTWP, as Jacqueline has already told us. And I want also to highlight the SME office, which is an office here in the agency that provides regulatory, financial, and administrative assistance support to companies that are designated as micro, small, and medium-sized enterprises. These are quite important in this, I think, in this context, because some of the alternative products are very niche areas of work, and they are being developed by small entities that don't have the regulatory sometimes experience, so they need additional support. And this is just, I think, quite a helpful graphical representation of all these supporting page. So you can see in the line from the almost the idea time until the authorization, and even beyond post-authorization. So having seen this, what are the challenges that are still there and that we should consider when we look at how we can support this, the availability of these alternatives? First would be, it seems quite simple, but the lack of consistent terminology, I would say internationally, of what is an alternative to antimicrobials, because per se is not a specific type of product. It includes different types of products. It could be quite important because then it could facilitate the harmonization of, if everybody knows what we are all talking about, then we can look at that and think of the requirements and harmonization of requirements overall. There are some suggestions there, products to reduce the need of the use of antimicrobials, or products that can contribute to reduction of the need for antimicrobials, or different combinations. But I think, the consistent terminology would be a point for consideration. The second one are the borderline products, and we heard an example from Luca before, which are those products that contain the same active substance, but it could be classified either as a VMP, veterinary medicinal product, feed additive, or biocide, depending on the intended use and the claims. And this is very important to consider the appropriate regulatory framework, so the standards, the requirements for authorization, and it's not only always that easy and that clear. So it's important to provide clarity to applicants, either from the agency, European agencies and national competent authorities. I refer to the current regulatory framework in place to regulate also, these products that can be considered as alternatives. So as we were saying, it provides a new framework since even before the implementation of the regulation up to now. For instance, the agency, the CVMP, have prepared scientific guidelines in immunologicals. More than 10 guidelines have been finalized and published, some reviewed, some completely new, to prepare for have the requirements in place when the regulation was applicable. Jacqueline again referred to scientific guidelines on novel therapies. One published in stem cells, bacteriophages, very close to be published. The guidelines in limited markets that I explained before, quality, safety, and efficacy of different types of products. But then, is that sufficient? Is that enough? Or is there a need for additional guidance, and specifically for different types of products, specific types of products that could be used as alternative for antimicrobials? And as Jacqueline said, sometimes we don't even know what is going to come in the near future. So it's important to know what the priorities would be and be ready for when developers are coming to us to at least inquire about the authorization of specific products. For instance, could be peptides, immunomodulators, gene-edited products, just an example. So the CVMP through working parties should be ready, and the agency to support these developers. Next one, promote all these measures we have in place, and I refer specifically to SMEs, which are small entities. Just promote all the incentives and the support available, because again, there's also very niche areas, and these entities don't have the regulatory knowledge. So it's very important that we promote them and we are visible for them. And this can be done through the next point, which is increasing the communication with the stakeholders on the development and authorization of alternatives overall. And the next one, which is foster the dialogue between the regulatory network in Europe overall, including industry, veterinary professional, and all the relevant parties. And that would be very important to identify, as we were saying before, the priorities. So looking at what diseases have the highest need for alternative treatments. For instance, again, Luca was referring to a specific one. And also what types of VMPs show the greatest promise. So it's going to be a priority to support and to be ready when they are ready to come for development, or even for inquiries about potential authorization. In relation to the international cooperation, again, we heard a lot about this morning, but we also heard that we always can be doing more, and in fact, there's more being done. So at the moment we exchange information, maybe we have to look at a higher level of harmonization of requirement, and specifically for global alliances for the support the authorization of alternatives. In relation to the measures already in place, I was referring to scientific advice, ITF. These are in place, but should be further promoted, and the support is there, and we can provide the appropriate forum to discuss the advice related to the development of these innovative products, and looking and thinking of priorities. Horizon scanning meetings with industry developers could be and would be very important, again, to know what is going to come next and to be ready to be prepared for that, the regulatory network. is there a need for further guidance? And if there is a need for further guidance in specific products, which one are going to be priority? Again, I know there's some repetition here, but that's important to highlight the main points. And for instance, would it be possible to have specific goals for projects on alternatives to antimicrobials to incentivize even more and provide some a bit of a push and stimulus on the development? And even more, could we establish some objective targets to evaluate and to monitor the success of the measures that are already in place, and then evaluate which ones could be needed to even strengthen the support to promote alternatives. So yeah, keep moving forward. And that was the last one. Thank you very much. And I have to say, any other ideas for overcoming challenges will be very welcome, and we are open to discuss. Thank you very much.
Thank you very much, Noemi. I think that showed that EMA already has systems and procedures in place for encouraging industry to develop these kind of products. But things are moving at a fast pace, and there's still a lot of regulatory and scientific issues that we need to address. So thank you for that. I think we have time now to take questions. So obviously we can take questions to any of our 4 presenters today. So I will throw the floor open to those people who are in the room first of all. If we don't have questions from people here, sorry, I see hands raised. So firstly, Ricardo, would you like to just introduce yourself and then go ahead with your question?
Yeah, good morning. Thank you very much for the presenters. I'm Ricardo Carapeto and I work in the Spanish Medicine Agency. And it's a question for Luca. It was very interesting, your presentation. And I was wondering, how do you deliver how do you carry out the fecal transplant? I mean, it's you just thoroughly deliver the filtered liquid into the mouth of the animal, or you have to gavage the animal, or just
you have to. We have gavaged the animal, so it was an intragastrical inoculation. But we are presently studying other strategies. So there is a lot to be done there, also in terms of modes of the administration. So optimize also the easiest and more sustainable way to administering the transplant to the piglets, because as I said, the handling individual piglets is quite complex and laborious. So, but to answer your question, they were inoculated intragastrically.
And an additional question: you mentioned that it's already done in the human side. How do they do that in humans? If you know about that.
Yes, they use capsules that are able to pass the gastric acidic barrier, and then they release the content in the intestinal tract. But to the best of my knowledge, this is the most frequent mode of administration. Double check, because I'm not a human doctor. And that could also be something to be considered, but how to administer a capsule to a piglet and be sure that the piglet will not chew it before it is ingested. That is another challenge to be overcome.
Thank you for that. And whilst, Luca, whilst you have the microphone, we had a question from Wolfgang Baumer. He says, very exciting presentation. Did the phages show any impact?
The phages?
Oh, have I got the wrong?
Did you get it wrong or he got it wrong?
Oh, sorry. Okay, we think it's for Jacqueline. Sorry.
Okay. In the preclinical trials, we had I mean, it's a long way to testing phages in a farm. We showed some good in vitro activity. I think the big challenge with phage therapy is the need, as it was well explained in one of the presentations, is that the pathogen is mutating all the time and becoming resistant to the phage. So you actually need to update the library. And this was done very well in the ex-Soviet Union. There were some hospitals, especially in Georgia, that were using successful phage therapy to treat hospital patients. It was a very resource-demanding task to keep the library updated. And then this can be done for a human hospital patient, but I doubt that there will be resources to make it sustainable in livestock production, also considering the issues on costs of food that I have mentioned at the end of the presentation. So I'm a little bit skeptical about the future of phage therapy in livestock production.
Thank you very much. So do we have any other questions from the room? Dr. Claassen, thank you.
Well, thank you very much, Helen, and thank you all for extremely interesting presentations. And I think it's really important that there is this focus on alternatives to antimicrobial therapy, because I don't believe that there's any incentive for new antimicrobials, real antimicrobials, to be developed for veterinary use, given also the fact that we have to reduce indeed that. I think it shows the diversity of approaches, and but also the regulatory constraints, that they have to fit within the regulatory framework. I think that's a recurring theme as well. So whether we don't have a specific classification for alternatives to antimicrobials in the regulation, they're either biologicals or pharmaceuticals, so they have to fit into any of these frameworks or novel therapies for that matter. Now, I'm very intrigued by Luca's presentation, because I think I see that more as a proof of concept, and I, as you mentioned rightly, it doesn't fit necessarily in any regulatory framework, but I could imagine that once you've established this proof of concept, and it is shown that it can replace that the gut microbiome can be analyzed, and you could even imagine that you analyze it to the extent where you do get a better understanding of what indeed it is in the gut microbiome that can sort this effect. Is it a combination of certain micro or certain bacteria in there? And to then subsequently, I know it's far away, but we're discussing the future, to produce that at an industrial scale. Now, I know that most of them are anaerobes, and they may be difficult to actually produce. But if you then come to a well-defined product, then it would fit in with the, I think, the definition of a veterinary medicine, which is medicinal products for veterinary use. So veterinary drugs or veterinary medicinal products are substances or combinations of substances to treat, prevent, or diagnose disease in animals. And I could see it being developed in the same way as probiotics, for example, as feed additives, that we got them well-defined claim, a well-defined composition, and that then would open up an avenue for using these also at larger scale, and maybe, as you said, use different application methodology as well that would avoid, the individual treatment the way it is and different ways. So I think it's very interesting, very promising approaches, but still a long way until it reaches an actual product. So I think the same holds true for a number of the other things that we've been saying. Phage therapy, we know where it is. I'm also intrigued by the nanotherapy opportunity. So can this be a game changer in the future? A long way to go. No question really, but just a remark I wanted to make. Thank you very much.
Yeah, and if I can comment on your remark, I completely agree. We are working on that, and I'm sure we are not the only ones at the University of Copenhagen. So to standardize, to find out, first of all, what are the beneficial bacteria that are having this effect. But the fear is that whatever we will do, even without considering the time issue, that this will take a lot of time, the fear is that this will never replace completely the role of an antibiotic. Because I see them more as tools to prevent, control the severity of the outbreak rather than curing an infection. So, and this is why I hope that one day, in addition to setting these benchmarking goals of reducing 50% and so on, we define what is the ultimate goal, what is the ultimate target that we want to achieve in the European Union. Because I think we have achieved already a lot. When I look at the graphs of the ESVAC, I still see a lot of heterogeneity. So maybe we need to adjust a little bit that. But talking about Denmark, which I think is one of the lowest consumers of antibiotics in relation to the total biomass that is produced, I can see that there is still this routine use at the two steps of the production cycle, birth and weaning, which is intrinsically linked to the way we produce the animals. I have mentioned the natural weaning time compared to the weaning time in intensive production. So we are creating these disease opportunities in the animals, which otherwise would not happen if the animals were brought up under different conditions. So maybe a way out, if we really want to eliminate or bring close to zero the use of antimicrobials in livestock, is to change the welfare conditions of intensive. I'm still talking about, I'm not one of those having naive thoughts about organic production in the field. So I'm still talking about intensive production, but under different animal welfare conditions. This would have a consequence on the price of the food products we buy. I said my opinion. Obviously, out there, the vast majority of the population have a different opinion, but we need to work on that and find out what collectively as a society we want to do in this situation.
Thank you, Luca. And following on from an earlier comment that you made about the fact that mostly these substances, alternatives, are probably going to be used to reduce antimicrobial use rather than replace it. We had a question from Anya Home, which we've received online. I think this is more of a regulatory question, so I'll hand it to Noemi in the first instance. So, will reduction of antimicrobial use be accepted as a benefit in the benefit-risk balance of vaccines in the future? Will it be accepted as a claim, for example, as part of an indication for vaccines? So, Noemi, thanks.
Thank you, Helen, and thank you, Anya. It's a very interesting question, not completely unexpected, but there's not that much I can say. Unfortunately, it's something that is currently, under discussion, and within the regulatory network, the Commission. There's not that much as I can say. I don't know if Eva wants to say something about that, or Eva, sorry to pass it to you.
No, thank you very much. And it's always very difficult to give a legal interpretation just coming like this, because we always need to check and to assess exactly what is the question. I do not think that a claim as such to reduce antimicrobials for a vaccine can be done, but of course, to give you so quickly a legal interpretation normally is not possible. But in any case, I mean, you can always send us any question of a legal nature. We are having a lot of discussions with the network, but also, as I said this morning, we are focusing a lot in interpretation questions, because it's not so straightforward and so easy to reply to all these questions, because we have to really give you a reply based on the legal framework.
Thank you, Eva. And Jordi Torren, I hand over to you. Thank you.
Yeah, thank you. Without answering the question, because as Eva was saying, I don't think we can answer the question. I must say I had also checked with the human colleagues if they were doing something similar, because that's a question that it's coming again and again to us. And it seems that it's not the usual thing to put, this kind of claim into the human products, and I guess that there will be a need to change substantially some of our legal requirements to allow a strict indication on antimicrobial reduction. Something else is that if it could be discussed, if there could be some kind of cite indication about what's been observed during the studies and things like that. But again, just to mention that the human colleagues do not have parallel indications on the human side. Thank you.
Thank you. I don't see any. Oh, sorry. Nancy, would you like to introduce yourself and then go ahead? Thanks.
Yes, thank you very much. Nancy Brienne, Federation of Veterinarians of Europe. Just following up on the previous discussion, I think indeed we will not see any new antibiotics in the field of veterinary medicine. So for that, it's very important to keep the old antibiotics efficient and on the market. And in that respect, we have some problems because we hear the Zoltan saying we have this nice AMEC categorization, every vet should hang it up in their clinic, which I completely agree with. But in some countries, they don't have the narrow spectrum antibiotics available. Well, they are available in other EU countries. And this is, of course, a frustration to these veterinarians. And if we could do something about that to make them more available on all EU markets, that would really be beneficial, I think, for the fight against IMR. One other frustration is the SPCs of the more older antibiotics. They differ between countries and they're not always updated. And sometimes the dose and the duration needs to be changed. And this is also legally not allowed. So this is another problem. But lastly, I want to say that I'm really happy that now the EMA will also start to focus on diagnostics tests to promote responsible use, because these tools can really play an enormous beneficial role. They're not miracle solutions. There are challenges with them also. And so it will be really nice to see how we can use them in the best way possible and what are the limitations also. Thank you.
Thank you for that. And whilst I think the diagnostic tests don't fall directly within the scope of the veterinary regulations, just to let you know that the CVMP's Antimicrobials Working Party is preparing or going to prepare very shortly a reflection paper which will look at the availability of diagnostic tests and how they can be used in the field. So do we have any further questions from members in the room? No. Oh, sorry. Yes, sorry, Carol, would you like to go ahead?
Thank you. Yeah, I'm going to abuse the fact that I've got the mic to ask two questions, very separate ones. The first one is really on, I wanted to, it's for Zoltan really, and the SVAC report. I think that report is seen, and my job is global, so as really from a global position as the Rolls Royce of reporting on use and stuff like that. And I think what was the latest number, 47% reduction?
That's correct.
Yeah,
yes.
Unfortunately, the counting only starts in 2018 for the 50% reduction. So still got about 30% to go or something like that. But what my question really was to you is, we don't, at global level, there is a similar report produced by WOA, not completely similar, but it's a good report which shows antibiotic use at global level by region, and certainly not as detailed. But what's available at European level on human use of antibiotics, and what's available at global level? As far as I know, there's very little in terms of an SVAC type report for antibiotic use in amongst humans, and the data that I've seen is showing quite large increases over the last few years, and no reductions like 47% or was it 38% in the States and different places. So I'd be very keen, and I say that because I'm very interested to know whether it goes to the transfer pathways point. I think one of you referred to the ECDC report. I think they said a couple of years ago, 75% of transfer they think happens in hospital settings or GP settings, human health settings, and the rest in everything else. So I'm very keen how we compare these things, and seeing as we're at EMA, medicines for humans and animals, I'm very keen to analyze if there is some attempt to analyze that kind of comparison in light of transfer routes and places. That was question one. That's for Zoltan. Question two, sorry, societal benefits and the point that was made. it's a fascinating discussion. Can you reward farmers who are not getting a financial reward for good behavior to do certain behavior? For example, how do you reward a farmer to use a methane reducing additive in their feed? If they can reduce the methane that's emitted from their animals by 20 or 30 or 50 or 80%, and these products are now beginning to get into the market, how is that farmer rewarded societally? Because that's a societal good, because he's reducing greenhouse gas emissions. Or how do you reward a farmer for using less antibiotics and using more of these alternatives or other products or better animal health or husbandry? Or how do you reward a farmer for making sure that the salmonella coming out of his products is much lower because he's vaccinated or certain things? These are interesting questions where there is no financial economic motivation for farmers to use certain products, but there is certainly a great payoff and benefits for society. So that's sorry, it's a rather large philosophical question, but and I know it's not in the regulation, and it probably will never be. But these are interesting questions to see how we societally can encourage people to do these good things and practice, which is of public benefit. Thank you.
Thank you. So Zoltan, if you'd like to take the first question.
Thank you very much. Yes, so EMA hosts SFAC, which is the surveillance network for collecting sales of veterinary antimicrobial products, in ECDC, the European Centre for Disease Prevention and Control, is the one who collects information on human consumption data. And of course, you can find all this information on the website. I agree that a lot more decrease was achieved in veterinary medicine. I would just like to encourage you to have a look at the JACRA report, which will be finalized soon, although we have to be cautious how we can compare antimicrobial consumption in humans and animals, but we still try to do that, and especially we show the trend of the reduction in the JACRA report. I think it's from 2014 till 2021, and yes, it's a clear reduction can be observed in veterinary medicine, but less in human medicine. I agree that more effort should be done in that area as well. Thank you.
Thank you. And then the second question that you posed, which was about how we can reward farmers for the work that they're doing, obviously, to tackle this issue. So I maybe would like to ask Professor Guardabassi about that one, because I noticed that you did some social research looking into the acceptability of the fecal transfers. And I'm just wondering, maybe you could give us a bit more information about the outcomes from that study.
I can answer personally, not as an expert. I'm thinking about some policies that have been implemented in Denmark, like the yellow card. I don't know if you are, Carol, are aware of it, which was going the opposite direction, meaning that those farmers that were using above the average, the amount of antibiotic use was above the average, were actually supposed to go back below the average. And there were a number of tools to implementing this, including financial costs for the farmer. I think if I was from the regulatory perspective, it is much easier to implement this approach than the rewarding one that that you suggested. But yes, sure, I think we need to consider tools, incentives for veterinarians and farmers to achieve the goals that we want to achieve.
Thank you. And then Ivo, please.
Yeah, in addition to that, I can support that, but this is not restricted to this specific field. This is something you see everywhere, ecological farming, other things. There's only so much that an economical market can do, and usually you need much more than coming from that. There's only so much what the passion of a person can do. You need more. And this is the greater good. So the entity responsible for the greater good is your national government. So one way or another, you can want it, you can wish it. In the end, you need legislation.
Thank you. So any other, sorry, Dr. Carapetto.
Yeah, I think that the current common agricultural policy has got something called eco schemes, something like that. That is like rewards that you give the farmer if you go beyond a regulation. And I think because those eco schemes are very well defined, where do you get the money? So I think this specific issue should be included in one of these things if there is a political interest. So just that. Thank you.
Thank you. And also Dr. Claassen.
Yeah, I noticed that Karel was asking a general question, but he's clearly soliciting to get some answers here. He wasn't asking someone specifically, Karel, but to give the view. I think because you mentioned the benefits to society, and then to me it's quite obvious that society should give something in return. It could be in the form of tax incentives or whatever, you know, assuming that they and I think we've seen something similar emerging on when we were asking farmers to contribute more to nature development and maybe not focusing on intensive livestock farming, but part of their fields were being transformed, you know, and doing it really in a different way to ensure that there's more diversity in nature, it's more attractive for wild birds, etc. So those are things that I think there's trends there. At the same time, and Professor Guardabassi said it as well, you know, the willingness of people to pay more or a realistic price for meat or animal products, I think that's one thing. But where we see that if you ask people in a survey, it's a well-known fact, they will say, oh yeah, I'm happy to pay 30% more, 10% more, or whatever. But when you look at the actual purchasing behavior, it doesn't happen. And in fact, a recent publication earlier this week in the Volkskrant, and possibly in other newspapers as well, what we see is a reduction in, let's say, alternatives for meat because they are more expensive, and people, because of crisis situations, fall back in buying what we call in the Netherlands kilo-knallers, so really cheap meat, where the cost of a kilo of chicken indeed is comparable to 1 liter of Coca-Cola. So it is the purchasing behavior of people that can ultimately change the system, but where it's not really happening because there's a cost associated with it generally, because you get lower yields and all you have to do huge investments in animal welfare. It's happening, but it's happening with indeed farmers that have that dedication to do that. Over.
So thank you, and thank you for ending this session with a very interesting question, a very interesting discussion. But I think now time is moving on and we need to go to the coffee break. So I'd just like to finish by thanking the four presenters today for their very fascinating presentations, and also thanking you, the audience, for the interesting questions as well. So before I close, I'll just hand back to Katarina so she can just give you a quick update on the rest of the schedule for this afternoon. Thank you, Katarina.
Thank you, Helen. Dear participants, just to inform you, as I have posted in the chat, the break. We will now break for coffee, and we will resume at 4 o'clock with 2 separate breakout sessions. The breakout sessions have 2 different links. They are copied, they were pasted in the chat. You can use, I would advise you to save it somewhere so you can swap between sessions if you want so. But the key message is that this session will end in 10 minutes. This broadcast will stop in 10 minutes, and then from 4 o'clock onwards, you should choose one of the 2 links that you have in the chat and follow the discussions there. Thank you very much.